Hello there, you're welcome. So this is some topic based questions extracted from past papers that is on topic 5. So I want you to try and see if you can attempt to them before listening to my answer. Of course, bear in mind that like I always advise my students, make sure you've actually gone through the topic 5 before attempting questions, whether it is past papers based on year or based on topic. So this one is based on topic 5. So energy and ecosystem. So the graph below, or the graph, shows how gross productivity and biomass in an area changed with time in the succession from bare soil to mature woodland. So you can see biomass there, and you can see gross productivity, you can see time there as it were. But this perforated line or dashed line is the productivity, and this is the biomass. Okay, so the question here is suggest the appropriate unit for the gross productivity. Okay, so looking at this, you can see time, you can see biomass. Then of course you should know there's a soil or there's a land on which this is happening. So the right unit will be the mass for biomass per area per year. Okay, that's because of the three things here. These two, time is quite really visible, but because of the soil or the land mass you are using, that's why per area. So when it comes to productivity, that's why that is in play here. Okay, next question. Explain the decrease in gross productivity as woodland matures. So looking at the graph here, you see that productivity is going up, then it starts decreasing at this point. So why would that happen? Even though it shows like biomass kept increasing anyway, so why will gross productivity decrease? So let's see that. So this is because as there are more plants, there is more competition for light. So that means plants would probably be some plants that are underneath will be shaded from photosynthesis, so their own photosynthesis will reduce. So as some will be shaded, as you can see that, so thereby reducing the rate of photosynthesis. So it's basically because there will be more plants. It's like you have plants at different levels. So some that are down there, so these ones on the floor will be obstructed from sunlight, so to say. So there will be gross productivity will reduce. These ones might increase, but it's going to affect some plants of the soil on the ground as it were. Okay, hope that made sense. So moving on. The next question says, use your knowledge of succession to explain the increase in biomass during the first 20 years. So when you look at that, like you're saying, this is 20 years, 40, 60, and other. So it means you're going to focus on something around here. So, and use your knowledge on that, and this is meant to be a 3 marker. Okay, so put that into consideration. So let's look at that. So really, the first set of organisms are pioneer species. So you should know that when there's succession on any, let's say this one is a bare rock. So the first set of animals of plants to start, we call them pioneer species. So the pioneer species will lead to formation of soil because this is a bare rock. So they will have to first, usually the guys we have, they are always lichen. That lichen is made up of algae and fungi, but don't worry about that yet. So this lichen we live in this harsh weather, and after they do that on this, not let me say harsh weather, this hard surface, it will then over time, this, please bear in mind that succession takes years to happen, like maybe 100 of years. So it doesn't just happen, it will take a long time for this lichen to die and break up the rock particles little by little by little. So the little soil particle plus their own dead matter will form this bit of humus, and that will now lead to increase in biodiversity, which means other bigger plants smaller plants can grow. Then after smaller plants have made the place habitable, bigger plants can then grow. Then you can go on and on until you have a bigger ecosystem as well. So you can see the first thing is you have to remember that pioneer species has to be mentioned. Then they would lead to formation of soil. Soil has to be formed. Then other plants can now grow. Then when there's smaller plant, you can have biodiversity, which means small insects can come around there, grasshoppers and things like that, and that will lead to more variety of plants and other things. Hope that made sense. Now, the next question says, use your knowledge or use the information in the graph and your knowledge of net productivity to explain why biomass shows increase after 100 years. So if you go up there, yeah, like this is 100 years, so biomass still increased. Yeah, like what does it increase on? So use your knowledge of gross or of net productivity to do that, not gross productivity now. So let's go to that part here. Now, this is because the habitat has reached its maximum carrying capacity. Now, please pay attention. It says little increase. I didn't make mention of that, please. Little increase, because you would see that the increase after 100, you see it was going up. Then the increase here is not as fast as it was before. Okay, so what happened was that the habitat has reached its carrying capacity. Sometimes we call it climax community. So as such, the net productivity equals to gross productivity minus respiratory loss. So organisms there, you have to, how do I put this here now? So you have to know that net productivity is the real productivity after you have subtracted gross productivity. Let me explain this. When you go to market to buy something, your net profit is really your profit after you have subtracted all your expenses from your gross capital and all of those things, so to say. I don't know if that explanation made sense, but the truth here is that in the context of productivity, energy, and ecosystem, your net productivity will be, you take away your respiratory loss, which organisms have used, from your gross productivity. So this leads to decrease in gross productivity due to what? Less photosynthesis. I mentioned earlier on that there is the plants, those bigger plants, will obstruct the smaller plants on the floor from, they will shade them off. I think I made mention of that in the questions before this. Okay. Next question says, suggest one reason for conserving woodlands: to protect the habitats, to protect endangered species, and to reduce global warming. So because the more plants you have, the more you can have more oxygen that would more oxygen in the atmosphere, and the plant that will absorb carbon dioxide for photosynthesis, so thereby reducing global warming. So any of this answer is correct for that. Okay. Now, chloroplast contains chlorophyll A and B. Scientists found tobacco plants with a mutation that caused them to make more chlorophyll B than normal tobacco plants. Okay, so that means these mutant plants, they will be making more chlorophyll B than normal one. They investigated the effect of this mutation on the rate of photosynthesis. Now, the scientists carried out the following investigation. They grew the normal and mutant plants. They grew some of each in low light intensity and grew other in high intensity. So you should know that we have two plants, the normal plant and the mutant plant. Both of them were grown in high intensity and also low intensity. Now, they isolated samples of chloroplasts from mature plants of both types. Okay, don't forget normal one and mutant ones. Finally, they measured the oxygen production by the chloroplasts they had isolated from this plant. So these were the results. You might have to pause the video here to really study this, because please truly pause the video here and see. So plants grown in light, in high light, that's the mutant one. Then the normal one grown in high light. So but these are both highlight, mutant and normal. Mutant plant grow in low lights, and normal plants grow in low light. So your it is highlight, low light, and it's both mutant first, then mutant first. So see the oxygen production. So trace that out, trace that out, trace that out. Okay, I don't want to make that too rough for you. So, but you should pause the video here and try to see that. So this is a 2 marker. So explain why the scientists measure the rate of production of oxygen in this investigation. So why was the rate of oxygen production what he measured? Okay, so try that. So the reason is because to measure the oxygen produced is is to show us the plant in a light dependent reaction. Then also know that the faster the oxygen produced, the faster the rate of light dependent reaction. So this is to, during photosynthesis we have the light dependent reaction. So this is to measure how much oxygen is being produced in light dependent reaction. Then the more the oxygen produced, the more we know that rate of light dependent reaction is fast, so to say. Hope that made sense. Don't forget that we have light dependent reaction, which happens in the day or in the presence of sun or light, and oxygen is produced. So we have to measure oxygen. Oxygen is a pointer to know that this is happening. And the more we have more of oxygen is to show that this reaction is happening fast. Hope that made sense. The next question says the scientists suggested that the mutant plants producing more chlorophyll B would grow faster than normal plants in the light intensity. Explain how this data support this suggestion. How many markers is that? 4 markers. That's a lot. So, well, this is because the chloroplasts for the mutant plants photosynthesized better at both light and low light. What's the proof? Let's see that real quick. See this. So see, the mutant plants at high light, it's higher. Oxygen production is high. At low light, it's still higher, because this is for low light for both of them. So when it was low light for both of them, it's still higher. When it is high light for both of them, it is still higher. Take note of that, okay? That makes sense. So it says here that, and that's because it photosynthesizes better at both high and low light intensity. As a result of that, there's more ATP and more NADP will be produced. Okay, I can say NADPH, okay, will be produced, because at the end of the day you have NADPH, or we say reduced NADP, or NADPH will be produced. This will increase the rate of light independent reaction, because these two guys is what you will use during your light independent reaction, which is Calvin cycle. So as a result, more sugar will be produced, which is essential for what? Respiration. The more the respiration, the faster the plant grows. The plant then synthesizes new and more organic matter from the excess sugar. So because the question is, why do we suggest that this guy will grow faster? You can see the answer here, because there will be, see the sequence, don't forget the flow. The fact that there's more photosynthesis happening because of the way you can see the graph shows that. Then once you have more of this, every other thing will be increased, and see what's going to happen eventually. There will be more plants, more organic, because there's more sugar produced. Since there's more sugar produced, there's more respiration, there's more energy to do anything, basically. Hope that makes sense. Okay, now you probably wonder, like, okay, why did I write so much? That's because if you stop your answer around here, you are still good to go. I just added extra stuff to make it more for you to understand. Okay, so when you send a question that has things like this, you can always use the same knowledge. Hope that makes sense. Next question says, scientists investigated the effect of mycorrhiza on the growth of pea plants with a nitrate fertilizer on an ammonium fertilizer. The fertilizers were identical except for the nitrate or ammonium. Now, before we move on, remember this word, mycorrhiza. When you hear the word myco, it means fungi. So rhiza has to do with root. So it's like an association between plant root and fungi, so to say. So the scientists took the pea seed, and sterilized their surfaces. They planted the seeds in soil that had been heated to 85 degrees Celsius for 2 days before use. The soil was sand and that contains no mineral ions useful to the plants. Now, explain why the scientists sterilized the soil, the surface of the seed, and then grew them in soil that had been heated for 85 degrees Celsius for 2 days. Why did that happen? So, Maka. Well, this is to kill any bacteria and fungus on the surface of the seed or in the soil, basically. So, as such, only the fungal added will have an effect. So, because I told you that mycorrhiza is associated between fungus and roots. So, the reason why the seed was sterilized and also the soil was to be sure that anything we want to see will be caused because of this, nothing else. Hope that makes sense. Explain why it was important that the soil contained no mineral ions useful for the plants. So think, why do you think that is important? So that only the nitrate or ammonium provided in the fertilizer will affect the growth. Because we said it here that these were made available, okay, for the plants, but there was nothing else we want to make available, and that's why we had to subject the soil to that. Now, the pea plants were divided into 4 groups: A, B, C, and D. Group A were treated with mycorrhiza and the nitrate fertilizer. Group B, mycorrhiza with fungus with nitrate fertilizer. So 3 things here: 1, 2, 3. This one is just 1 and 2. Mycorrhiza, this one is heated. 1 mycorrhiza added with ammonium, not nitrate here. Okay, so the next one was mycorrhiza fungus added with ammonium fertilizer, but it is not heated. So it's quite a funny one, but how many markers is 2 markers? So explain how group A and C act as control. How does A and C act as control? A and C, how did they act as control? Look at their variables, look at the things there. Okay, to show the effect of nitrate or ammonium. So, because if you notice now, C and A here, what is that word again? Okay, so you can see they are both heated here. Mycorrhiza fungus, then you can see the fact that the only variable here is what? Ammonium and nitrates. Okay, hope that made sense, so that the effects of the fungus can be seen. So that's the reason why they have those things being identical. So don't forget, it says explain how A and C act as control. Hope that made sense. Okay. Now, after 6 weeks, the scientists removed the plant from the soil and cut the root from the shoots. They dried up the plant material in an oven at 90 degrees Celsius for 3 days. Then they determined the mean dryness of the root and shoot of each of this group of peas. Talking about those peas that agree with these things. So suggest what the scientists should have done during the drying process to be sure that all the water had been removed from the plant sample. Okay, let's just think about that. So what they should have done is the sample should be weighed at intervals during the drying process, so you can be sure that the thing is actually drying. Really, is it reducing or it's not reducing, so to say? So to see if the weight becomes constant or not. Okay, so that is the idea of the whole thing. So as you're drying from day 1, day 2, day 3, there should be you should be weighing it so that you are not you are sure that there's no you are not amassing more weight, or the plant is really drying up gradually, so to say. That's just basically what it is. Okay. Now, the next question says the scientists' results are shown in the table below. You can see the heated plant and the one in fungus, heated fungus, and the one with nitrate fungus. This was without, just basically what you saw here. Basically, that's what you have in the table here. But see the result. In the root, but pay attention to basically these figures, like those figures. Basically, those figures. That's what. So, what conclusion can be drawn from the data in the table about the following: the effect of fungus on plant growth? So, look at the one that has fungus there, and look at the weight of the root and the shoot. Take a moment to look at that. Really, there should be something out you can see from there. Well, the fungus increase the growth of shoot and roots in both plants. Can we see that? See, that's quite high. 1.6, 9.8. I mean, compare that with the remaining. That's quite really high. Then, more growth is seen in one with nitrates. How do we see that? So the one that has nitrate has more growth. Can you see that? There's quite clearly more growth. Yeah, there's more in both of them, but this is more. Okay. Then, and that is, excuse me, before I move on, that is, let me get something right here. That is in, oh, sorry, I think I missed something before I move on. Now, I think there's a mistake I made in the first one. So look at this. Please pay attention here. It's not a big mistake, so there's nothing much. So the one that has fungus, which is this one, and this one, you can see that it is high in the roots, high in the shoot, high in the roots, high in the shoot compared to the other ones. Number one. So that's what this first one was saying. Now it says it's now more growth seen in the one with the nitrate. So if you compare, this is the one with ammonia fertilizer, but see 0.96, 4.10, but compare that with the ones. So that means the one with nitrate is still more than the one in the ammonia. Hope that made sense. Okay. Now state the effect of the nitrate fertilizer and ammonia fertilizer on growth of the pea plant. Well, the dry mass for both are similarly lesser compared to B and D. Well, we can see that anyways, because if you see the one in fertilizer compared to the nitrate fertilizer compared to the ammonia fertilizer. So if you look at the one of ammonia, they are actually, I hope I've not switched it the other way around now. Okay, compared to B and D. Yes, so this B, hold on one sec. So yes, so this is what it means actually. The dry mass for both are similarly lesser compared to B and D. So if you look at this one and this one, A and C is both smaller in every way compared to B and D, because they had fungus added to them, really, and not just fungus added to them because of I'm trying to remember the other factor there. Because it was not heated, because this were not heated actually, so they were not heated because this one had heat added to them, so to say. So that's something I was going to say, point out there. So, but they were similarly lesser, so there's no much significance as standard deviation overlaps. Okay, the next question says the scientists determined the dry mass of the root and shoot separately. The reason for this was they were interested in the ratio of shoot to root of pea plants. It is the shoot of the pea plant that is harvested for commercial purpose. Explain why determination of dry mass was an appropriate method to use in this investigation. Why is the dry mass used, and why is that reliable? Okay, so the dry mass measures the increase in organic material, which you want to see, as this shows the true mass of the shoot. You remember from your basic GCSE, even in A levels that the dry mass of an organism is the true mass, is stable. So water content tends to what varies. That's why we're not interested in that, because water content can vary with season, can vary with anything, so to say. Now, okay, which treatment gave the best result in commercial terms? Justify your answer. So, which results from what we've observed give the so it says B and C fungus containing fertilizer. Now, let me see that. So yes, so B, I mean the fungus with nitrate containing fertilizer. So what I mean by that is this one, B, like that seems to be the best in terms of everything. Look at the ratio of the root to the shoot, or the shoot to the root, as the case may be. So if I go back there, so as this give the largest shoot to root ratio, it has the largest dry mass of shoot being 9.8, and that ratio will be, if that's going to be 6.01 ratio 1 compared with the ammonia content, which is going to be 4.18 versus ratio 1, if you get what I mean. So like, what did I do to do that? So I found the ratio of this against the ratio of this. So that's why that's what basically I did. So ratio of this is what you saw here, which is 6.9 ratio 1. That's shoot to root. Then for the ammonia containing is 4.18 ratio 1. So that's the reason why this is quite more. That's plant B. Hope that made sense. Okay. Last question, if I'm not mistaken, says upwelling is a process where water moves from deeper parts of the sea to the surface. This water contains a lot of nutrients from the remains of dead organisms. Nitrates are two of these nutrients. They provide a source of nitrogen and phosphorus for cells. Give a biological molecule that contains nitrogen and phosphorus. Really, this should be a lot of examples you should remember from this one. A lot. So we have the likes of DNA, protein, amino acid, NAD, NADP, RNA, ATP. Then those are contains phosphorus. Basically, it's almost the same thing. The only thing that's different is the GP and TP that you have as part of your substrates in glycolysis and the likes. Glycolysis and the likes. So hope that made sense. So that these are the examples of substances that contains nitrogen and that. Describe the role of microorganisms in producing nitrate from the remains of dead organisms. How many markers is that? 3 markers. So what's the role of microorganisms? This might be calling your attention to one of the cycles. If you remember those cycles. So, and that is what about your nitrogen cycle, really. That's what I'm trying to call your attention to. So the action of saprobionts. Saprobionts are organisms that cause things to decay, such as bacteria and fungi, which causes decay of dead organisms. As a result of the decay, the protein from this dead organism is broken down into what? Ammonia. Ammonia is then converted into what? Nitrites, and then nitrates. Nitrite is now converted into nitrates by nitrifying bacteria. So these are something you don't want to forget, really. Very important. Okay, I think at this point I should make mention of the fact that if you are doing EDUCAS, ammonia to nitrite is nitrosomonas. Nitrosomonas. While nitrite to nitrate is Nitrobacter. Please pardon that, it can be very confusing actually. Nitrobacter. Okay, so ammonia to nitrate, that is done by Nitrosomonas. Then nitrite to nitrate is done by Nitrobacter. Those words can be very, very hard to pronounce. But like I said, this is relevant only to those that are doing EDUCAS. AK doesn't bother really about that. Okay, hope that makes sense. And now the next question says, upwelling often results. One second, let me be sure we're done with that. Okay, so upwelling often results in high primary productivity in coastal waters. Explain why some of the most productive fishing areas are found in coastal areas. Why? Okay, and that should be, let me reveal the answer. That should be because the more nitrates and phosphates you have, the more you have more producers, which producers like what? Like algae and plants will grow. So more producers lead to more food for the fish, thereby making more fish to reproduce. So it's just because of the fact that you have a lot of these two guys, plants will grow. And in this case here, the first guy that will grow is going to be the likes of algae, so to say. Okay. And once you have a producer, you can rest assured that other organisms will grow there. Then the question here says, nitrates from fertilizer applied to crops may enter ponds and lakes. Explain how nitrates may cause the death of fish. Basically, you should remember this talking about what? Eutrophication. Basically, that's what it's trying to say there. So this is quite sorry the way I wrote it, but this will cause the eutrophication. This will cause eutrophication, which involves massive growth of algae, which is called algal bloom. The algal bloom will cover the surface of the pond. This reduces light availability for photosynthesis, thereby causing the submerged plant to die. Now, once that happens, saprophytes will use up the oxygen in the water for respiration while decaying the plant. So because they want to decay the plant that has died, and they will end up using oxygen, less oxygen means there will be less oxygen available for the fish, and the fish will eventually die, basically. And that explains eutrophication. And sometimes people will say that the plant, what's it called again? The algal bloom will shield off the amount of oxygen available also, but really that's not a challenge because they are submerged plants. But now that the submerged plants have died due to photosynthesis, then the whole thing will take its effect, so to say. Hope that made sense. All right, I'll see you in the next.